big words, so let's repeat them together. Bacterial transformation.
Bacterial transformation.
And protein purification.
Protein purification.
What bacteria is. Okay, does anyone want to take a shot at what it does? Yeah?
Me?
Yeah.
I think it's like something that something that goes into like water and like turns it so it's not like good to drink, right?
Yeah, that's what bacteria can be. What about you?
There's bad bacteria that can cause infections, but there's also good bacteria that actually help to live.
Yeah.
I actually know about that because once I had an infection, so that's how I know about that.
Oh, very good. What about you?
Bacteria helps the environment stay alive because without any bacteria, everything would be dead. So bad bacteria for you might be good for something else.
Very good. All good answers. They're all true, actually. Bacteria can be good or bad. It can be in circles or squares or ovals, and it's in every living thing. And so it's in water, and it's in plants and people. And so bacterial transformation is when you're taking one piece of bacteria and you're turning it into something else that you want it to be. So in your table groups, you each get an envelope. So you guys can open your envelopes up and each define a red circle.
Red circle.
Okay, so we're going to go through bacterial transformation, and this is how we do it in the high school, but instead of paper cutouts, we have real materials. So we have real pieces, so these are pieces of DNA. They're little circles, they're called plasmids. And what you're going to do with these plasmids is I want everybody to find a test tube, which is this item that was in their envelope. So what we're going to do next is we're going to take this test tube that has our bacteria, which is the green stuff in it, and the plasmids, which are the red circles, and we're going to take our little blue bucket. Our little blue bucket, we're going to pretend it has steaming hot water in it, okay? And so we're going to take our test tube, and we're going to put it in our bucket. Okay, so the bucket has hot water in it, right? So in the test tube with all of the little DNA pieces, what the hot water does is it mixes the DNA and the bacteria, so it all becomes one big thing. So we're going to take our test tube away now. We're going to take it out of the water. So now this is all mixed up together, okay? So next, what we're going to do is we're going to put it on a plate. So this brown stuff on the plate is what the bacteria likes to eat. So think of your favorite food, like spaghetti or grilled cheese. Bacteria, they like to eat something called agar, which smells really bad, and it's like gooey, it's like slime. It's like hard slime. So people don't eat it, but bacteria love to eat it. So we're gonna take our DNAs, the ones from our tube, and oops, while they were in our tube, they mixed up. So this is it after it's all mixed up. So we're gonna put our bacteria on top of the plate so it can eat and it can grow. After it's all done growing, we're going to put it into a test tube so it can get all mixed up and warm again. So in the test tube, after it's done growing, what you're going to do with the test tube is the test tube has a special chemical in it called buffer. And the buffer is what separates the materials in the bacteria. So once you have your bacteria in the test tube, it'll all come out, and you'll get a whole new set of DNA and a whole new set of proteins. Okay, good. All right, so who thinks they know what protein purification is? Just like if you break it down, what's protein, what's purification? Does anybody have any ideas? Yeah.
So the protein would be like at first it would be good to eat, but then you have to purify it and take out the bad stuff and keep in the good stuff.
Really good, yeah. Anyone else? Yeah?
kind of like water purification, where you put it in a bowl and then heat it up, and then the bad stuff goes up, and then the good stuff is down still.
Yeah, very good. Awesome. So that's right on the money, you guys. That's so good. So protein purification is a lot like what you said. You're taking the cell and you're taking the protein that you want. Like if you want your hair to grow longer or your nails to be stronger or you want to grow more muscles, you take that specific protein out of the cell and then you take it and you use it for what you want it for. So we have another activity. This one I'm very excited for. So everybody gets an egg. What color is everyone's egg? Just think about it. My egg is yellow, right? Your egg is purple, your egg is blue. Your egg is blue. Okay, and so inside our egg, we have, this is our cell, right? So this is the cell that we're going to be working with. This is a cell from our body, right? And so inside the cell, there's a whole bunch of different proteins and there's a whole bunch of different plasmids. So we're trying to do protein purification. So we're trying to take the protein that we want out of the cell, okay? So inside your egg, there's lots of different colored pieces of paper, okay? And so if you have a yellow egg, you're trying to just get the yellow pieces of paper out, right? If you have a purple egg, just the purple, blue, just blue. And I want you to do this, and I want you to try and not get any of the other pieces of paper out, okay? So you can use your fingers and just pick out the right color, but this is going to simulate what protein purification is. So scientists use this in their labs, and they open the cell, and they find the protein they want, and they extract it, or they take it out of the cell. So, you guys can start.
I almost took a yellow out.
Scientists. So, going back to the activity we did a little bit earlier with bacterial transformation, who remembers what the first step was? Does anyone remember? Oh, in the back? Yeah.
Put the red dots over the what's called into
The test tube? Yeah. Awesome. All right, who remembers step 2?
Then you put the test tube in hot water,
Yes, and do you remember why you put it in hot water?
so it can warm up.
Almost. That's part of the reason.
It mixes.
It mixes. Awesome. All right, how about step 3? Yeah.
Figure out which put your like thing in your
So
science.
close. You have the green part right. In the back of the blue shirt.
Put your take the test tube out of the hot water.
Nice. Yep. All right, step 4. Right there.
You put like the slimy stuff in the science.
Oh, you're so close. Where does it have to go to grow before you put it in the tube?
Oh, in your container.
Yep, on your plate so it can eat and grow big and strong. Who remembers step 5? Yeah.
Put it back into the test tube.
Oh, close. Which test tube? The one with the kind of fat bottom or the one with the skinny bottom? Yeah, yeah.
That bottom.
I know, so funny.
He's so polite he didn't even want to say the word fat.
So, step 6, what do you have after you put it in the test tube with the fat bottom? Yeah?
Put it, you
Forgot? That's okay. Yeah?
you have a new set of cells and bacteria and proteins.
Yes, you do. Do you remember why or how it's important that you have new proteins and new cells? Yeah?
Because then you can put the like these in like pills.
Very good.
And
That's so good.
and then there's like eggs.
Yeah, awesome. That's right on the money, you guys. That's so good.
Stellar, thank you.
Yay!